Here we will learn about PCR technique. PCR, the full form of PCR is polymerase chain reaction. Here the polymerase is the name of an enzyme. In the DNA replication process, we all know about DNA dependent DNA polymerase. But in this technique, DNA dependent DNA polymerases or eukaryotic DNA polymerases cannot be used because here we will use high temperature, and in high temperature they can be denatured. So instead of using eukaryotic DNA polymerases, we have to use Taq polymerase. This Taq polymerase is isolated from a hot spring bacteria. The name of this bacteria is Thermus aquaticus. Now, chain reaction means it is rapidly grown or exponentially growth is occurred in this technique. This technique was invented by Dr. Kary Mullis in the year of 1983, and for this reason he got Nobel Prize in the year of 1993 in the field of chemistry. Now, PCR technique means simply to amplify the DNA sequences or increasing the number exponentially. It is an in vitro process. In vitro process means the method is done within the test tube in a laboratory condition. In the other hand, in vivo condition means the process is done in the biological system, in the plant or animal body. For this PCR technique, primer design is very important. We should not use too short, in the other hand too long primer for this reaction, because if the primer is too short, then it will attach in many places of the template strand. In the other hand, if the primer is too large, then it will take more time to attach in the template strand. So we have to use an ideal primer that should be 18 to 25 base pairs or nucleotides long. And obviously the primer should be single stranded. Two primers are added in two template strands. At first we have to take a double stranded DNA, then we have to denature it, and then we have to annealing or addition of primer is done. The nature of the primer is also very important. It should not be self-complementary. There should not be self-complementary bases. If there is self-complementary bases, then it will join with each other and form hairpin loop. And if hairpin loop are formed, then the primer is saturated and cannot react with the template strands. Here a primer sequence is 5' TAG TCA TGC ATGA CG C3'. And here a lot of complementary base pairs are present, so they attach with each other. Here adenine is joined with thymine by double bond, and guanine is joined with cytosine by triple bond. We all know this, and forms hairpin loop. So the primer is self-complementary and can be saturated with the help of the self-complementary bases and cannot react with the template strand. So if we have to use an ideal primer, the primer should not be self-complementary. Now for the base composition of the primer, GC content should be enough rich, that is 40% to 60%, and the remaining should be 80 rich. Because the GC pairing is stronger than AT pairing. So what happens? The primer should be strong enough to join with the template strand. And the bases should be evenly distributed for pairing. That should be follow the Chargaff's rule. That means the adenine and the thymine ratio should be same, and the guanine should be same amount according to cytosine. Now coming to the process, there are 3 steps. The first step is denaturation, the second step is annealing, and the third step is extension. In the first step or denaturation process, we have to take a double-stranded DNA. Then we cannot use DNA helicases. Helicase can unwind the coiling and separate 2 strands from each other. But in higher temperature, helicase is also denatured, so we cannot use helicase. Hence, we have to give higher temperature, approximately 94 degrees centigrade, and the DNA strands are denatured, and what happened? 2 strands are separated from each other. One strand is 5' to 3' and another strand is 3' to 5' here shown. After that, for the amplification of DNA strands, primer annealing is needed. And for this reason, the temperature should be approx 50 degrees centigrade to 70 degrees centigrade or approx 54 degrees centigrade. And what happens? 2 primers are inserted in both the template strands. There should be a little bit of temperature changes in both the primers, approx 5 degrees centigrade different, and this temperature is called TM temperature. The different temperatures in both the primers, approx 5 degrees centigrade. Now in the third step, what happens? Extension happens. And the temperature should be 70 degrees centigrade to 75 degrees centigrade or approximately 72 degrees centigrade. And here we will use Taq polymerase, and for the buffer Mg2+ is used. Because DNA polymerases can be denatured in such higher temperature, so we have to use Taq polymerase. And Taq polymerase is isolated from the bacteria. The name of the bacteria is Thermus aquaticus, and it is an hot spring bacteria, Thermus aquaticus. Now Taq polymerase is used, Mg2+ is used as buffer, DNTP is also used. But what happened? The extension happens, but the proofreading is not happened. Why? Because Taq polymerase has no proofreading activity, and we know that dependent DNA polymerases have proofreading activity, but Taq polymerase has no proofreading activity, so we have to use TTth DNA or TTth DNA pol, which is isolated from another bacteria, Thermophilus. Otherwise, we have to use TLI DNA polymerase, and it is isolated from another hot spring bacteria, Thermococcus litoralis. So, we can use TTth DNA polymerase, otherwise TLI DNA polymerase for proofreading activity, because proofreading activity is necessary for DNA replication. Now what happens? Extension happens and 2 strands are formed. Now this process is done repeatedly for 35 to 40 times for having more and more DNA. And after having such kind of DNAs, for analysis, agarose gel electrophoresis is used, or labeling the PCR primers or nucleotides with fluorescent dyes. Otherwise, staining of the amplified DNA product with a chemical dye such as ethidium bromide is used for analysis. The whole process is done for the maintenance of temperature in PCR machine. This machine is also called thermal cycler. So simply what happens? At first we have to take double-stranded DNA, then we have to denature it with high temperature, then we will get 2 strands, and then DNA or primer annealing is added. Primers are added in both strands. There is a difference of temperature, approx 5 degrees centigrade in both the primers, and that is called TM temperature. And this primer annealing is done in the 54 degrees centigrade. And after that, extension is done, where we have to use Taq polymerase instead of DNA polymerases. Mg2+ is used as buffer. TTth DNA polymerase is also used for proofreading activity. And then we will get complementary strands. This process is goes on until we get specific amount of DNA. Now the DNA amounts will be doubled or amplified in each cycle, that is 2 to 4 to 8 to A16, and in ideal condition the increasing formula should be NF equal to Ni into 2 to the power n. This formula is very important in case of ideal condition, where NF denotes final amount of DNA, Ni denotes initial DNA amount, and small n equal to number of cycle. If small n equal to 2 or number of cycle is 2 and Ni or initial DNA amount equal to 10, then final amount of DNA or NF should be 40. Here is another formula, but it is applied if the efficiency would not be 100%. Then the formula should be NF equal to Ni into 1 plus percentage efficiency whole to the power n. This PCR process is used in Human Genome Project, diagnosis for diseases, clones and sequence genes, detection of pathogens, and forensic medicine to identify criminals. So what we learned is that the primer is very important in this process. It should be 18 to 25 nucleotides long and should not be self-complementary or saturated. And the base composition of the primer, where GC content should be enough or 40 to 60%, and the bases should be evenly distributed. For the process we have to take double-stranded DNA, then we have to denature it with higher temperature in the thermal cycler, and then we anneal it or add primers in both the strands, and then extension happen in 72 degrees centigrade, where Taq polymerase is used for extension, TTth DNA polymerase is used for proofreading, Mg2+ used for maintenance, the buffer, and the extension happens, and hence we get more and more DNA exponentially. This is a short elaboration of PCR technique. Hope you like it.